Hi friends, so now I am back with my next video, and in this video I am going to talk about complementation system. Before going ahead, please make sure to like, share and subscribe my channel. So what is complementation system and where it is used? So the complementation system is mainly used in genetics. As I have talked to you in my last video that in genetics we have genes which controls for protein, which codes for protein, and these proteins are the functional unit and it produces a phenotype. So complementation system is used to check if there is one phenotype which is controlled by two genes, okay, and if the genes that controls the phenotype, if they have mutation in the same gene or in two different gene, mutation in same gene or two different genes. So what does that mean is, suppose you have a phenotype which is controlled by two genes, and because of any mutations you are not getting that phenotype. So the question is where that mutation is. Is it in the same gene or is it in the two different genes? Because as you know that each gene is represented by two alleles. And as I have talked about the dominant and recessive phenotype in my last video, so you know that dominant is something which is the allele is hides the function of the another one. So as I have talked about the phenotype height, and suppose you have a phenotype height which is controlled by a gene or alleles which are represented by TT, and in the heterozygous condition it is like this, and in the recessive condition it is both small. So this is homozygous dominant, zygous dominant, and this is heterozygous dominant, heterozygous dominant, and this thing, the TT, is homozygous recessive. So what complementation system uses is this homozygous recessive. So what does that mean? That the mutation is present only when the gene is in homozygous recessive condition, which means that the alleles both are in homozygous recessive condition, and if that is possible, the phenotype that we get is the mutant form. Otherwise, in homozygous dominant and heterozygous dominant, it will be in the wild type form. Okay. So now we know that it happens only when it's the homozygous recessive. So I will show you by an example. Suppose that we are talking about Drosophila, okay? Because in genetics most of the studies were done on Drosophila, and suppose we are talking about the phenotype eye color, okay? So the eye color generally suppose is white, and this phenotype is controlled by two genes. Like if the gene A is present, okay, A is present, it causes some chemical reaction or some changes, and the phenotype is still white, okay. And then there is another gene which is present, okay, which codes for protein B, and this protein B changes the color of the white to red, okay, or green or whatever you want to say. And so you know, if there is any changes, if there is any mutation in A, it will still be white, and if there is mutation in B, it will also be white, because B cannot function without an A, and A cannot proceed further to red without B. So now we want to know like the by these, suppose if the parent or any individual have still white but not red, so we don't know if the mutation is in B or is in A. So this test is to check whether the mutation is present in A or is in B. So how we can do it? Suppose we have one parent in which the mutation is in A. So as I have told you, the mutation is there only when it's in homozygous recessive condition and the B is normal. And this parent is crossed with another parent, the mutation is in B now. And so the AA will be there and then the BB. And then the offsprings from this parents will be. What do you think will be? So we get A from here, the capital A, right? So what we get is A and also the B from here, okay? Okay. And then we get from this parent small a and capital B. So now because the two genes are in heterozygous condition, we get the wild type phenotype, right? Which is red color. Okay. So from this we know that the mutation is in two different genes because the offspring that we are getting is red in color. What happens if the mutation is in same gene? So like you can see here, instead of using this mutation in one, the mutation is in both the genes. And then if it is crossed with the parent which is normal, so which is like these, okay, and the offspring that we can get from here is capital A, small b, capital A, capital B from here, and small b, small a and small b from here. But because the mutations are still in different genes from this parent, we still get the heterozygous one and wild type phenotype, okay? And the third case in which the mutation is in the same gene. Suppose the mutation is in A. Okay, so what do you get from here? So suppose there is this parent in which the mutation is in small a both the time. So there it's small a, capital B, and it is crossed with the parent which the mutation is also in the capital B. So from here what we get is the offsprings that we get will be small a from here, capital B from here, small a from here, capital B from here. And in this case, because A is in homozygous recessive condition, so the phenotype that we get is white. So we can say that they are in the same gene and they are not complementing each other, because if they were complementing each other, that they are in different alleles or in different genes, and one allele is enough to show this expression. But by this we can say that they are in the same gene and they are not complementing each other to produce the wild type phenotype. So I hope this is clear, and if you have any doubt, please let me know in the comment section. Thank you.